Can people hear me?
Hi, G. Yeah, we can hear you good.
Okay. Okay, I think it's already 2 past the hour, so let's get started. So hello, everyone. Welcome to DCMB seminar series. Today, we are pleased to welcome Dr. Xiaotian Zhang, a very special guest from our own medical school. Dr. Xiaotian Zhang received his Ph.D. from Baylor College of Medicine, with his Ph.D. thesis focused on the role of DNA methylation synergy in leukemia development. He was previously the Andel Special Postdoc Fellow, working on the 3D genome in leukemia. He's now a research track faculty in Pathology Department. Dr. Zhang's research focus on the epigenetic regulation of key pathogenic genes in leukemia, particularly on the high-order chromatin structure in disease. He has a number of high-profile publications on Nature Genetic, molecular cell, and blood. So with that, I ask you to give your full attention to Dr. Zhang and help me welcome him to the virtual stage. Dr. Zhang, you are on.
Okay, thank you, Jie. So, so it's a great honor to be here and then to get invited by my friend Jie, and also we're actually doing collaboration now. But anyway, so I will just get started. And can everyone see my slides?
Yeah, looks good.
Okay, thank you. So okay, so thank you for Jie for the invitation in the first slide, and then I will just try to talk about our recent work on the 3D organization of human genome in the normal and the malignant hematopoiesis. By saying this hematopoiesis, I mean I'm actually meaning is the production of the blood. So okay, so then I will just start from a broader view of the non-coding genome. So as we know from the finish of the human draft genome, we know that most of our genome is actually non-coding, and then these non-coding elements actually in the decades of research has been proved to be very functional in regulating important gene expression, and is having a lot of genetic variants and associated with the disease. So so in that case, to study this non-coding genome, and we know that a lot of the time these non-coding genomes lies pretty far away from the protein coding genes. And if with the traditional reverse or forward genetics method will take a very long time to actually discover these elements. And luckily, actually, in the decades of epigenomic research, we know now these non-coding, these like long-range regulatory elements or the non-coding genome elements actually can be found by the labeling of the by the certain marks in the epigenetic marks. Like histone marks, and also some like insulators were looking for the CTCF sites. So that's why NIH actually initiated ENCODE projects and to look for the transcription as well as all this genomic modifications in the human tissues and cells. So today I'm mostly going to talk about is the 3-dimensional chromatin structure, as well as the histone modification and DNA methylation and the correlation between them. So, and here is all the type of the next generation sequencing technologies, and it's highlighted by ENCODE. I think in this audience, probably I don't need much time, to introduce this. So, firstly, I'll start I'll start on the method on how to study the 3D genomic structures or chromatin structures. But we know that actually in a linear genome, and then if we want to know the distribution of certain epigenomic mark, so we simply align the sequencing data, and then just it's sort of like a beads on the string model, and we know where it's distributed. However, for the 3D genome, it actually has been folded and into the very small nucleus. So what is the principle for such folding? And people has developed a method and pioneered by Decker about 2 decades ago called chromatin conformation capture. So this, the idea about this method is basically assume that two locus, which actually are distant from each other in the linear genome, will actually come together in this three-dimensional chromatin structure, and then maybe held by a certain protein. But the way we do it is actually we take the cells and fix the cells to preserve such organization. And after we preserve this organization, we perform the restriction enzyme digestion, and then to leave the close by locus in the 3D genome together. And then we perform the proximity ligation. And then in that case, the locus close up in the 3D space will form these chimeric molecules. Then it will be identified in the next generation sequencing. So, and to visualize such interactions and bioinformaticians have been using something as called correlation matrix, and to show the relationship between the interactions between different locus. You can imagine if there's only the linear DNA, and so when we perform this kind of like adapter ligation, so this proximity ligation, so most of the locus will associate with themselves, and sometimes by chance you will have some interactions with this neighboring regions. However, if you perform this kind of like proximity ligated chromatin structure, and then you will find like the locus like A and D will have significantly higher frequency to interact in compare with the random interaction by chance. So this is how we actually, this is a method how we're going to use and to study and to visualize the interaction between the locus and the genome wide. And in the real data sets, however, we don't do this kind of like restriction fragments. There's no such perfect restriction fragments can do and can be aligned perfectly. So the bioinformatic researchers has been doing is actually they take the genome and then they chop the genome into very small bins, so containing several locus. So in that case, and then they also use this correlation matrix to visualize such correlation between the different locus of the genome. And so as you can expect, so most of the interaction comes from the interaction between a locus and its nearby locus. In this case, you're generating this kind of like a diagonal signal mainly. However, sometimes, for example, like the genes from, like the locus from C to F, you actually form this correlation between each other. And then this kind of closely correlated region is called a compartment in the 3D chromatin structure. So this kind of compartments, and then when researchers are looking at this kind of compartments in the genome level, whole genome level, what they find out is actually the genome is actually organized into this mutually exclusive compartments. But it means actually for the region from A to I, they may form like in two compartments, which actually separate from each other. And then from here, we can have two compartments and containing the locus A to D and I to I. And then in the middle is something we call insulators. So this particular insulator is separating the two compartments. So then depending on how you bin the genome, so if you bin the genome in really large scale, then you can get this something called AB compartments. So A compartment is something is active and then it's enriched and it's a gene-rich region. So the B compartment is silent and gene-poor region. And if you further zoom in, you can get this topological social domain, which spans from 0.1 to 1 megabase. But still you can see, and they are actually mutually exclusive. And sometimes you will see it's actually this kind of like insulator, this insulator of elements will form very strong interactions on top of the topological social domain reads. And then this particular interaction is something called chromosome loops. So And so this is basically the types of interactions in our genome, and people has been widely studied. Is actually topological social domains and also the chromosome loops showing here. So all I have been showing you is all based on the sequencing and chromatin confirmation data, captured data. And what is the physical force or biochemical force to mediate such formation of the domains and loops? So it turns out to be people find out is we need a true complex molecular complex here to form this topological social domain, which is actually cohesion complex and then CTCF sites. So what it does is actually, we know that cohesion is a molecular motor. So, and here comes the cohesion, and cohesion will actually extrude the chromatin fibers, and what CTCF do is actually it will stop the cohesion extrusion process. And then, and interestingly, and the CTCF sites have to be in a certain orientation. And so, and it has to be in this kind of like convergent head-to-head orientation. And then this extruded part, and here is actually the topological social domain. And then the CTCF, which stop the cohesion extrusion, lies on the border of TADs and also the loops. It's called the tidal loop anchors, and which actually is CTCF binding sites. So this is basically people's idea now of how this TADs and loops forms. So we know, okay, so these TADs and loops forms. And what's its function in regulating the gene expression? So we find, so there's multiple literatures about this. And then what people has been find out is actually these TADs actually enhance the long-range interaction between an enhancer and a gene inside of the TADs. So some people is also arguing that actually the TADs is preventing the enhancers to assess another gene in another TADs. There's several literature recently argue against this model. It's still like hotly and debated. And so, what I'm going to talk about today is actually about the generation of the blood cells in our body every day. So basically, we're really interested is actually the normal hematopoiesis process is called, and you can see in this what hematopoiesis mean is the production of blood. So as you can see here in this chart of the normal hematopoiesis, is actually on the top here is the stem cells is going to always maintain itself and then give rise to the precursor and progenitors that will later turns into all these mature blood cells. Let's say, the red blood cell carry oxygen, and then platelets stop the bleeding, and also, all the granulocyte and monocyte fight for the bacteria, and also the lymphocytes that generate antibodies. And so, as you can see here, and this is actually the process in our body is happening every day. And you can see here, this normal process is coupled, and from the stem cell to the mature cell is called differentiation. As you can see from this differentiation process, so the nucleus of the cells actually undergoes a huge alteration in the morphology. So, and so I was trained as a hematologist. So we are always wondering, okay, so in this process, so what exactly has been changed and during this process to promote this kind of like proper differentiation. So bearing that in mind, so the model we choose is actually red blood cell differentiation. So in the high school biology, we know that the mammal red blood cell is without a nucleus. And then, and during this differentiation, and at first, the red blood cell precursor progenitors, they still have a nucleus. But during this process, the nucleus become very small and then condensate, and then condensate. And so it's actually really interesting to see. So what during this nuclear condensation process, so what's the TADs and loop level is changed. So based on this idea, so then we isolate the hematopoietic stem cells and also the red blood cell precursors, and then perform this high resolution in situ high-C method. Try to dissect, the 3D chromatin architecture change during this red blood cell differentiation process. Okay, so and here I will show you some of the data. I mean, that's very raw data, and so as you can see, it's actually showing here in upper triangle is the stem cell high-C contact map, and then the lower triangle here is the red blood cell precursor high-C contact map. So in the globally, what we find out is actually the TAT level doesn't change that much. As you can see in this plot, it's also the same thing. So the TAT precision also doesn't change much as well. And also we find a very interesting part is actually in the red blood cell precursors, the loop number has been decreased significantly. As you can also see in this plot, the what arrow pointing is the 2 chromosome loops. However, after differentiation, these loops disappears. And but we do have some difficulty, in correlating this kind of like loop disappearance with the gene expression. And also, this is the same thing happens on the TATs. And then so make us a little bit disappointing as the first time when we first look at the data. And also something we discovered is all people has discovered before. However, I mean the TAT and loops are not the only structures you can find in this high-C heat map. And then so today I'm going to talk about the 2 things. So first thing is actually we have discovered that actually one kind of epigenetic elements called demethylation canyons. They actually are interacting with each other, form a very distinct 3D genome interactions in the hematopoietic stem cells. And also, if I have time, I'll talk about briefly about our collaboration program with Jie on the AML 3D genomics. So, okay. So I will first, then I will just start it from our part 1 on the discovery about the demethylation canyon interactions in stem cells. So if some of you work on demethylation, you probably know the demethylation is the modifications occurs on the CpG site, which actually is this primary role believed to be repress the transcription. And so most of our genome has been methylated. So every CpG sites in our genome is almost methylated, 100%. So in this whole genome bisulfite track, and then these sites are all like very heavily methylated to 90% or 80%-ish. So here is the dip. So this kind of dip in the whole genome bisulfite sequencing is actually the CpG islands, and which is a cluster of CpG sites is not getting methylated. And then and this is a part of the genome. And then if you actually scroll around and then to look for the different regions, you will see actually sometimes you will encounter this really wide region actually is devoid of any DNA methylation. So this type of the dip in the whole genome bisulfite sequencing is what we call demethylation canyons. And then here's one of the example of the biggest demethylation canyons in the mouse hematopoietic stem cells. As you can see, and this actually spans around 40 kilobase pair. And then we defined the demethylation canyon as the unmethylated region, so span over 3.5 kb. And then that's actually enriched, and we find out it's important, and it's also enriched for the developmental homobox genes. So, okay, so, and then just by serendipity, then we actually, we load the demethylation canyon onto the high-C map. What we find out is actually, now these demethylation canyons are actually form this very long-range interactions outside of this like topological social domains. As you can see in this example, is actually the mutual interactions between DRX1 locus SP9 locus and also HOXD locus. As you can see, they each form and this form this mutual interactions. So suggesting all these three locus actually cluster together. And you can also see that this is the interactions between these demethylation canyons are actually stem cell specific. And after the differentiation into the red blood cell precursors, these interactions has been gone. And if you take a look at the fish data and then to further validate this, you can also see that in the stem cell, the two locus are clustered together, and in the red blood cell, however, it's getting separated. So so when we find this, we find okay, this is something very exciting because this is something people haven't been described before. Rather, and because our emphasis has been mostly on the tags and loops during this differentiation process. However, at that time we already know is actually the demethylation canyon is actually not is a mixture of in other words it's actually heterogeneous in the chromatin state. So it means it's actually a mixture of active enhancer promoter and also the polycomb binding sites. As you can see here, it's the active promoter, and then here is the polycomb binding sites. Because these are the two chromatin modification histone marks for antagonizing the demethylation in our genome. So then here pops up the question. So what specific demethylation canyons are interacting? So you may guess essentially it's the active part, because the active enhancer promoter may form this kind of like very strong interactions promoting the gene expressions. So we decided to use a method called the aggregation peak analysis to look for such interactions. So how this method works is basically take your regional interest and then its flanking regions, and then tile up these interactions in this 2D contact matrix. If you have your region interest is actually interacting, then you can see this type of, and then you will have a peak and also in this kind of like heat map. And it's over, and you can see there's significantly signal increase over the background. So if it's not interacting, the center will be roughly the same as the background. So this is how we actually tease out such significant interactions between your region of interest. So here's one example of the chromosome loops. So as you can see here for the chromosome loops, the number here is just indicating how far away the two loop anchors are getting separated. But first, you can see that if the two loops, the anchors are separated by around 200 kilobase pairs. So there's a significant interactions between the two loop anchors. As you can see, the signal comes from in the center and then is significantly higher over the background. And also you can see here is that so the size of loop means the distance between the two loop anchors, so seldom exceed 2 megabases. So because the genome in our genome we have many CTCF and cohesion binding sites, so making this kind of loop size smaller than 2 megabases. And then when we first perform this analysis on demethylation canyons, so what we find out is actually most of the demethylation canyons are not interacting. So this represents 80% of all demethylation canyons. So categorized by the length of the canyons. And interestingly, is actually we find out it's only longest 20% of canyons is actually having the having the length over 7.3 kb interacting with each other. So this just indicating that, okay, so, and also you can see that it's actually no matter how distance the two canyon locus are separated from each other, you can always see such strong interactions. And in one extreme case, we even find that the two demethylation canyons are interacting interacting with each other. And then these two locus are actually separated by around 130 megabase. So this is almost the entire long arm of the chromosome, of human chromosome 2. So this suggests that, okay, so the demethylation kinase interaction is distinct from chromosome loop interactions. And also, as I said previously, so the loop interactions have a feature about the CTCF orientation, where it's formed these convergent interactions. So as you can see here, for the loop anchors, so they will have a lot of this head-to-head convergent CTCF interaction. However, for the long kind interactions, actually does not favor any types of the CTCF orientations. Further suggests that actually these long kind interactions are independent of the cohesion loop intrusion model. We have other data to show that upon the degradation of cohesion, and there is no change in the kind interaction as well. So further just showing that the long kind interactions and cohesion interactions are two independent 3D genomics features in the human genome. So then what makes this longest 20% kinase special? So and I showed you before is actually these kinases are actually enriched for K27 trimethylation and also for the active K27 acetylation. So then we take a look at whether these long kinases are enriched for the K27 trimethylations or the K27 acetylations. So what we find out is these regions are actually in the genome are the strongest regions for the K27 trimethylation enrichment. And while at the same time, it does not have any overlap with enhancer with the active marks. So I mean, then I think this make us very confident to name these special long kinase grand kinase. So okay, so then because the K27 trimethylation levels in these grand kinase are so high, so then let's come to a question. So whether these are the forces driving the formation of these grand kinase interactions. So we know that this K27 trimethylation is deposited by something called polycomb repressive complex 2. And to be specific, this K27 trimethylation is methylated by EZH2. And then now we have a very good EZH2 inhibitors in clinical trial. Then, so our idea is actually we treat the cells with EZH2 inhibitors and then see whether that will change the 3D genome, the 3D interactions of these grand kinase interactions. So as you can see here, if we treat with EZH2 inhibitors, it's very effectively eradicate the K27 trimethylation globally, and then in the Grand Canyon regions as well. And when we perform the aggregation peak analysis, we find it's actually the treatment of EZH2 inhibitors has reduced the Grand Canyon interactions from 73% to 22%. And then at the same time, the loop level is not affected. So suggesting that the high K27 trimethylation level in Grand Canyons are indeed required for such interactions. So here is our model. So we think is actually so the demethylation, the unmethylated, the unmethylated region just serve as a reservoir to actually allow the K27 trimethylation nucleosomes and to be deposited. So in a certain stage, I mean, if it hits a certain level, let's just say Grand Canyon level, and then you have enough K27 trimethylation nucleosomes, it will probably segregate by themselves or recruit PRC1 protein called Win1b, and then may form this punctus. And upon the EZH2 treatment, so you lost the K27 trimethylation. So they may lose this self-segregate property, or they may lose, the recruitment of the Ring1B, and then and you can see here after treatment, the Ring1B, all goes out of the nucleus. So suggesting that, showing that it also maybe lost the recruitment of Ring1B to form this kind of clusters in the 3D genomes. So, so without that, I'll just summarize my first part. So we have been shown that actually demethylation canyons, only the longest 20% of canyons are interacting. And these are exclusively polycomb targets with the highest K27 trimethylation level. And it's also cohesion CTCF independent. And then we published this thing on Molecular Cell in May, and as a cover article, and as we depict the Grand Canyons as the glacier here, holding a lot of interlinked K27 trimethylation nucleosome here. Okay, so here's my first part. So I would like to stop here and then just to see whether you guys have any questions. Okay, so any questions? No? Can people hear me?
Yeah, we can hear you.
It was very good. One thing I thought, I don't know if this is a question or observation, but the canyons, I think, indicate a place where you might have not so many transcription factor proteins that bind into the chromatin. And so, it's probably allowing the chromatin to close down to a heterochromatin state and be more repressed. So, it's just, I think it's really good. I mean, the talk so far is great. I mean, we're learning from you. It's really good. And it does lend itself to the idea that likely we're going to be able to machine learn against the, the chromatin capture, data, especially as we have, collections of that data, to look for these subtleties and these different lengths of loops and some of the transitions that would occur in that space. It's thinking more of it as a phase diagram than something, the way we do in physical chemistry. So, I just think it's really good. What you're doing is great so far. So I don't want to monopolize it, but we are alive and paying attention.
Oh, okay. Okay. Thank you. Thank you. Yeah.
Okay.
Okay.
Yeah. Okay.
Okay. Okay. Then, yeah, I think I definitely agree with you. So, we probably just, these subtle interactions sometimes will bury, in the tags and the loops. And then this is also something, when Jie helped me to analyze the data, and this is something can be buried.
Well, another thing is just in terms of the timing of these different things. I mean, the sequence and how fast certain transitions are, like the extrusion of the loops and, that kind of thing. I mean, there's a time series evolution here that I think, we lose sometimes when we look at an averaging over, because the chromatin capture, chromatin of the problems, of course, is that the chromatin capture itself is an average of a lot of different chromatin states. There are different chromatin states in different nuclei, and it's not a single cell technique, but it's an averaging population technique. So we have to, so we have to be aware of that too. So there's that limits, somewhat what we can do. But, with Jia on one side of you and Josh on the other, you're probably going to be able to figure that out too.
Okay. Yeah.
Yeah.
That is
very good.
Okay. Thank you. Okay. So I'll just, keep going. So, okay. So I'll show you all this Grand Canyon interactions. So, so then what's the function of this thing in regulating gene expressions? As you know that it's, for enhancer promoter interactions, it's actually really straightforward mechanism where you have enhancer and then promote the gene expression. But in this case, it's the GRS enhancer controlling the sonic hedgehog locus. So what will happen is actually if you don't have the GRS locus, then the sonic hedgehog will not express. And after it's not expressed, the mouse do not have a lag. So it will turn into this snake-like mouse, according to this author of the papers. So for the Grand-Canyon interaction, it's a little bit different, right? So because we are talking about the repressive to repressive canyon interactions. And interesting thing is most of the Grand Canyons are actually enriched for the developmental important homeobox genes. But if we use CRISPR-Cas9 to remove some of these canyons, you always impact the gene. So then it makes such interrogation of the non-coding sequence very difficult. So then what about if you have a canyon without a gene in there, so then you can easily dissect this canyon by using CRISPR-Cas9 technology. Sorry about that. So we are very lucky is actually we do, it's also by serendipity, we actually find a locus like that. So we name this thing called genus canyon because it does not have a gene in there. So this particular canyon is actually forming interactions with the important developmental and stem cell gene, HOXA cluster genes. As you can see here, the canyons are forming this kind of like interactions, and also they both enrich for high K27 trimethylations. And you can also see that this kind of interactions it disappears after differentiation, suggesting it's a stem cell specific interaction. And also interesting thing is actually in between the posterior HOXA and also the genus canyon is the active, there's 3 classical TAS domain, and then also 3 chromosome loops that connecting the active HOXA with its upstream enhancers. So then you can actually thinking is that, so this is a cartoon to show this thing where the repressive canyons are interacting with each other on outside, while the enhancer promoter interaction and chromosome loops are nested inside. And within here is actually the active HOXA genes are both the important transcriptional factors for the stem cell genes, and also the repressive one is also important developmental regulators. So then, because the genus canyon has no gene in there, so makes its gene editing very straightforward. So then we just design 2 guides and then to directly cut off this region, and then after that, then we'll just assess how much stem cell and how much stem cell activity is there, and how much differentiation is there. So here we're using 2 flow cytometry to assess this. So we're using CD38 as a differentiation marker, and the CD34 as a self-renewal marker. So it means it's more stem cell-like. So as you can see here, in the control guide treated group, so there's a 40% of cells is still maintain its stem cell marker. And however, if we delete the genus canyon, there's only 15%. So and also for differentiated cells, and after the deletion, so and now we have 2-fold increase in the differentiated cells. And also when we test the stem cell activity by colony forming assay of the genus canyon deleted cells and also the control cells, you can see that the colony forming activities in these genus canyon deleted cells has also been greatly reduced. Then what about the gene expression? But as I said before, the active HOXA genes are all the important stem cell genes, and then the repressive HOXA genes are actually expressed in very low level. And then when we you may think is actually because this is actually repressor. So after the remove repressor, you may affect this repressor gene for them to highly express. However, when we do the QRT-PCR, we find out is actually this repressed HOXA11 and HOXA13 genes has not changed expression that much. So what has been really impacted is actually this active HOX genes, which actually driving these stem cell self-renew. So that actually just means so the genus canyon is actually not a repressor to repress these genes, but also it maintains the active HOXA expression. So then we also perform the 3D genomic analysis after the deletion of genus canyons. As you can see here, previously we have really good interactions between the genus canyon and also the posterior HOXA genes. So after the deletion, this interaction has been apparently lost. And then we also take a look at these enhancer promoters between the active HOXA region. And then we find out is actually for these enhancer promoter loops, and then after deletion of the genus canyon, this loop interaction has also been significantly reduced. So, so in that case, we propose a model is actually the repressive interactions between the genus canyon and the HOXA regions are actually forming as a stable scaffold to allow the proper enhancer promoter interactions. So when you lock such interactions, the enhancer promoters are not be able to form these interactions and very well. So in that case, the active HOXA gene expression will be impacted. So this is actually very similar, to the to people thinking in the task model, where the structure is actually the task structure is allowing the proper interactions between these enhancer and promoters to enforcing these interactions. So we're thinking maybe, in this scenario, in this HOXA locus, this genus canyon and HOXA also forming this type of structure to maintain the active gene transcription. So, okay, so I think this is my second part. And then I think we still have a little bit of time. So then we'll just switch gear a little bit, trying to talk about the 3D genomics of acute myeloid leukemia. So as I showed you before, so this is the normal hematopoiesis where, it's characterized by the, it's actually also characterized by the heterogeneity because, in a normal cell, in a normal condition, our body is trying to produce all kinds of, diverse cells to maintain our body properly. However, in the leukemia case, and this is something called leukemia blast or leukemia cells, you can see these leukemia cells just look like each other, and then they are actually very resemble the early undifferentiated cells. So this is the feature about leukemia, because the uncontrolled proliferation, and it's also a differentiation block. And then these kind of uncontrolled proliferation and differentiation block and can be promoted by the expression of these HOX cluster genes and many other genes that actually can promote such transformation. So one interesting thing about the acute myeloid leukemia is actually the acute myeloid leukemias are highly enriched for the DNA methylation associated mutations. So you can see there's mutations in DNMT3A, which is actually the enzyme directly add the methyl group onto the DNA. And then TAT2 and IDH1 and 2, when they are mutated, they will also cause the hypermethylation. You will see that these mutations are actually very significantly more frequent, in AML in comparison with all the other cancers. As you can see, the other cancers does not have like very frequent mutations in these demethylation associated genes, associated mutations. So, so why we're thinking this demethylation mutation is important, because actually we are studying the demethylation canyon. So I've been telling so far is all the function about canyons, and also the histone modifications in the canyons are important for interactions. So what about the demethylation? So whether the demethylation, a change, in the leukemia process may also alter the 3D genomic interactions about these Grand Canyons. And here's some introduction about IDH and TAT mutations. So as I told you, as I said a little bit, so essentially IDH and TAT mutation will cause a DNA hypermethylation. So how does the IDH and TAT mutation will cause that? So this is the pathway actually has been discovered about 10 years ago. So what the IDH and IDH in the wild type condition do is actually produce a compound called alpha ketone glutarate in TCA cycle, and then alpha ketone glutarate is an important substrate for the TAT the DNA demethylates and also the histone demethylates to get rid of the methyl mark from the DNA. However, when the IDH get mutated, and then this mutation will overproduce something called the 2-hydroxyglutarate, which actually looks very like alpha ketone glutarate, and then will compete with alpha ketone glutarate and inhibit the function of TATs and then KDMs. Then in that case, it will cause the fail of demethylation and then causing the hypermethylation globally. And also it will cause the histone hypermethylation globally as well. So basically speaking, when the cell is mutated with IDH or TATs, so it will cause the global DNA hypermethylation, will increase the hypermethylation in the CPG sites. So we wonder whether we can mimic this situation by treating the normal stem cells with 2-hydroxyglutarate. So after we treat the 2-hydroxyglutarate, we find out is actually it's also eradicates the Grand Canyon interactions. Now it's down to 12%, so it's even more, and then the than the EZH2 treatment. So suggesting that okay, maybe the hypermethylation caused by D2G also cause the loss of interactions. So then how this thing can happen? So we showed that actually before. So the demethylation canyon is modulated by DMT3A, so the enzyme that actually adding the methylation. And after you knock out the DMT3A, so your canyon actually getting expanded. And then if you knock out type 2, now you get hypermethylation in the canyon. Now your canyon has been really become very small. So it may be actually this region after you get hypermethylation, it squeezes out the ketone 7 trimethylation, and then making the interaction a lot weaker. So also because since I mentioned about the IDH and TAT mutant, so then we decide whether we want to look at in the primary patient sample carrying IDH TAT mutation, whether there is some 3D genomic change. So then we actually begin to profile the primary acute myeloid leukemia and some cell lines at the same time, and then we have this covers MLV enrichment leukemia, NPM1 mutated leukemia, and also RUNX1 leukemia. And in NPM1 mutated leukemia, and we have IDH mutation and TAT mutation here, and then here is the 3D clustering of these cells based on their 3D genomic profile. As you can see, these cell lines are all the kind of like outliers in comparison with these primary cells. And we decide to look all at the primary cells. So then the next thing we do is just to compare the topological associated domain structures in this AML. And we find out is actually these topological associated domains in the leukemias and also in the normal stem cells are pretty much alike. So as you can see, that first of all, the test number doesn't change much. And also the precision of tests doesn't change much as well. And then sometimes you can get this, the tag alteration events. Sometimes one tag in the stem cell can become 2 in the leukemia, and also vice versa. And then we're also investigating how that can contribute to the gene expression. And the interesting thing we find, and then here come to the 3D genomic analysis on the canyon interactions. So we take out the region about the of the Grand Canyons in the CD34 stem cells. Then we just see whether these interactions are still there in these leukemias. So we find is actually for this leukemia that's not involved in any type of the epigenetic mutations, the canyon interactions are pretty much maintained the same or even stronger. And then when we take a look at the and in this ML re-enrichment leukemia, it's becoming decreased. The interaction decreased a little bit. And then in the IDH and TAT mutation, however, the Grand Canyon interaction just further goes down. So this may suggest that actually IDH and TAT mutations may cause a hypermethylation in this Grand Canyon region, and then cause the loss of such interactions. So here's the example, and then you can see that in ML rearrangement leukemia, you still have some of the HOXD and then DLX1 interactions suggested here. That in the IDH1 mutated leukemia, then these interactions between the Grand Canyons has been clearly eradicated. So this is further just to show you guys is actually IDH and TAB mutations may cause the DNA hypermethylation, and then all that can also eradicate these 3D interactions between these Grand Canyons. Okay, so we also find out is actually for the important genes that driving leukemia formation, and they also have this leukemia-specific hyperacetylated region. And this hyperacetylated region somehow form its own chromosome domain in comparison with the normal stem cells. As you can see in PBX3 locus here, and also the ZAP2 locus, where you can clearly see a new domain has been popped out in the leukemia. And then importantly, these are all the genes that actually directly promote the leukemia development. And so I will just skip one slide here, and then here's our summary of what we find in the acute myeloid leukemia. Is actually in the normal hematopoietic stem cells, we have very strong interactions between the polycomb-mediated Grand Canyons, Grand demethylation canyons, and then there's maybe some, enhancer promoter interactions in some of the key oncological, oncogenic genes. However, in the leukemia case, now we actually lost such interactions between the polycomb-mediated demethylation canyons, and at the same time, so the enhancer and promoters has formed their own domains, and then driving a high expression of leukemia of the key oncogenic genes like ZAP2, PBX3, and HOXA, which I actually don't have time to show you. So it's sort of like the seesaw, not just like has been tilted. Okay, so with all that, I would like to acknowledge my former team, and especially Haley, who perform all this next generation sequencing library construction, and also our wonderful collaborator, Dr. Jie lab, and then and also Fan and Xinjun Ziqiao, who help on this analysis, and also my current lab head, and also my funding source. And here's the QR code for you guys to assess the AML 3D genomics data. Okay, so that's all for my talk today. So I'll just stop and then take questions. Thank you. Yeah.
Okay, thank you so much, Xiaotian. That's a fabulous talk. So
okay,
first, let's see whether we have questions from the audience. So if you have a question, you can unmute yourself.
It's been said that cancer is a disease of the DNA. I think you're suggesting that maybe it's a disease of the chromatin. I mean, did you want to comment on that?
Yes, I'm sorry. I think yeah, I think in leukemia case, actually, the chromatin definitely, involved a lot, in developing leukemia. So like classical case, like ML rearrangement leukemia, is something definitely people has been used a lot to start the transformation. And I mean, I don't know why, but I think in the solid tumors, these chromatin associated mutations are not that frequent. And then you can only find like MLL3 in some of the cancers, but it's not like chromatin associated mutations are not as frequent as the like TP53 or RAS mutations in all these solid tumors. So I mean, it's definitely, I think in my field, leukemia seems like a very unique case where the chromatins, and it's definitely very important. And also demethylation is something like also very, very important, because it is the case where you can see there's a lot of demethylation associated mutations. Yes.
So I'd like to follow up. It's Gil Uman. Very nice talk. 2 points. First is, are you aware of the AML work under the CPTAC Proteogenomics program of the National Cancer Institute. Arul Chanayan and Alexei Najvitsky and others of us are involved in those cancer-specific studies.
I see.
Is that relevant to your work?
No, actually, I'm not quite sure about this work. I've been mostly working on genomics. So this proteomics is pretty, I mean, we haven't heard much about this. Yeah.
That's okay, but here's the reason I'm telling you about it, is there's a new strategy about all this, which is to bring the genomics and the proteomics people together.
Yeah.
We call it proteogenomics.
Oh, that's great.
So
Yeah.
I would encourage you, when you have the chance,
Yeah.
to, at least look up the papers from Najvitsky and Chanayan.
Okay.
We're in your department.
Okay.
And some of them I'm on also. It's very interesting about various kinds of cancers. And we've not been so much involved about AML, but other groups around the country have, including Brian Drucker in Oregon Health Sciences Center. I'm sure you know that name.
Yes, that's true. Yeah.
And the other is about the TADs.
Yes.
So there's a lot of interest here in the TADs in Indika Rajapakse's group. Do you know Indika and his students like Stephen Lindsley and Scott Ronquist? Did you know these folks?
No, I haven't actually. Yeah.
That's why we like to have these seminars, to try to connect people.
Okay.
Here in Michigan and other places, of course, also, but especially here and in our department. So if you could take those names of Indika, Indika, Rajapakse, Rajasp, Rajapakse, and Stephen Lindsley, Lindsly. We're doing some really nice projects there, and they're deep into TADs. I think you'd find it very interesting. There's also a whole seminar series just starting. A famous mathematician who's interested in TADs.
Oh, that's great.
And that's Stephen Schmale. That's a name worth knowing. He has 4 degrees from the University of Michigan.
Wow, that's
Fields medalist, wonderful man, just celebrated his 90th birthday in July with a big symposium.
Wow, okay.
And we are part of the Schmale Research Institute here at Michigan.
Okay.
Right, which has a meeting tomorrow between 1 and 5, Gil, so we shouldn't forget that.
Well, I thought it was at 4 o'clock tomorrow. It's all afternoon?
Yeah, I think it's all afternoon. So check that out. So this is really good. So have you maintained your contacts with the Van Andel Institute?
Yeah, I do have some contact with them.
Good. I think that should be maintained. That's good. And that
they have some participation in the CPTAC also. CPTAC is,
that brings you back and connects you in with TGen in Arizona. I think it's a good thing to do. And we build up a lot of collaborations with those guys early on, and then fellow, I'm trying to remember his name, who's the dean over there. So I went to school with him, so we've got a good connections. So so my next question, I guess, would be, that the chromatin capture and some of the methodology that throughout your talk that you really showed some nice work with, hasn't, I think, been able to be used kind of as in a clinical validation setting. It's a basic tool, it's a tool for basic research, but it isn't a tool for kind of diagnostic or
this is
I wonder how you can what you have to do to move this into the translate it into something that could be directly applied.
Exactly. So I think this is something so actually I have a slide, but I think it's too much, so I take it out. So I think HOXA has been validated to be a very good tool to dissect the structural variations. So this is something I also try to collaborate with Jie with, because when the structural variation is generated, it would generate something very distinct from this tags and loops. So and when this thing generated is actually you can immediately catch that. So that's why I think this is a very good tool to actually identify different types of structural variations, and then and also what some of my work is actually show is that if you actually can downsample these things just to maybe 10 million or 20 million reads, and then do this HOXA, you can see these translocations super easily, and then maybe some of the deletions and also some inversions as well. So I think there's definitely something for this high-C technologies, we can make this thing into to starting like the new structural variations. Actually, in my case, we do find actually in some of the AML samples, there is a very novel deletions actually people haven't been discovered before, and that's apparently cover important tumor suppressor genes. So this is definitely something if we want to go that route, we still need some computation assistance. So I think you guys probably know this thing better than me, but but apparently I think this is a direction we can go to clinical part. Yeah. Yeah.
Okay, it's already 3 minutes past the hour. I think we should stop here. Thank you again for this wonderful talk. And thank you for attending our seminar. I remind you next week we will have another 4DN talk from Gemma from CMU. So hope to see you again. Thank you.
Okay.
Stay tuned.
No, it was really good talk. I think it was
Thank
really
you very
great.
much.
And I hope you can work with us and collaborate with our department.
Okay.
And
Thank you
follow up
very
on
much.
all these things that Dr. Owen was saying. I think there's really a lot of opportunity, and I know you will with the GF who you're working with already. So it's really a good thing. Thank you so much.
Okay. Thank you so much.
Thank you.